everyone. Today we will continue on chapter 2, which is on the topic of cellular respirations. So, however, on this week we only focus on the 2 out of 3 stages of cellular respiration, which is glycolysis and citric acid cycle. So, what is aerobic respirations? Aerobic respiration also referred to cellular respiration, which is it is a catabolic pathway that consume oxygen and organic molecules which refer to the glucose, and thus it will produce carbon dioxide, water molecules, and also energy. Okay, in ATP. So the breakdown from one molecule of glucose will produce about 30 to 32 molecules of ATP. And aerobic respiration is the most efficient catabolic pathway and is carried most in the eukaryotic cells and also in many prokaryotic organisms. And the aerobic respiration or this cellular respiration have 3 stages. The first stage is known as glycolysis, second stage Krebs cycle, and the third stage is oxidative phosphorylation. So glycolysis is the breakdown of the glucose, which is the organic molecules, into 2 molecules of pyruvate, and it will produce ATP through the process of substrate level phosphorylation. The second stage is Krebs cycle, and it is also known as citric acid cycle or tricarboxylic acid cycle. And this process completes the breakdown of the glucose and produce ATP by using substrate level phosphorylations also. And the third stage is oxidative phosphorylation, in which it involves 2 stages, which is electron transport chain, chemiosmosis, and ATP synthesis, and thus it produces ATP through oxidative phosphorylation. So where does the cellular respiration take place? They take place into 2 parts of the cell, in which glycolysis in the first stage, the glycolysis, which is the glycolysis, it occurs in the cytoplasm. Meanwhile, Krebs cycle and oxidative phosphorylation occur in mitochondria. To be specific, Krebs cycle occur in the matrix of mitochondria, the blue color over here. Meanwhile, oxidative phosphorylation take place in the inner membrane of the mitochondria. So in eukaryote, cellular respiration occur in cytoplasm, which is the glycolysis, and also in mitochondria involving Krebs cycle and also oxidative phosphorylation. However, prokaryote cells do not have mitochondria. Thus, the cellular respiration occur in cytoplasm and plasma membrane of the cell. So this is the overview of how does the cellular respiration occur and take place. In glycolysis, one glucose is converted into 2 molecules of pyruvate, and it will enter the mitochondria through transition process. I will explain about this one later. And the pyruvate converted into acetyl-CoA, and it will enter Krebs cycle. It will produce ATP. Electron will be transported by NADH and FADH2. All of it will go through oxidative phosphorylation, and this process also produce ATP by using oxidative phosphorylation process. So what is glycolysis? As I mentioned earlier, glycolysis is the breakdown of glucose that occur in cytoplasm. And glycolysis can be happen into 2 conditions, either with the presence of oxygen or without the presence of oxygen. So in the presence of oxygen, the glycolysis function at the first stage of respiration, in which the glucose, which have 6 carbon skeleton, is oxidized, becoming three carbon molecules of pyruvic acid or pyruvate and produce two molecules of ATP by substrate level phosphorylation, and it will produce NAD plus by the process of reduction. So pyruvate later will move to mitochondria where it will completely oxidize to become carbon dioxide and water in the Krebs cycle process. So there are two phases in glycolysis. We have energy investment phase and energy yielding phase. And it can occur whether or not there is oxygen present. So look at here, glucose converted into two molecules of pyruvate. If there is an oxygen, pyruvate will form acetyl-CoA and thus goes to citric acid cycle and etc. Meanwhile, if there is no oxygen present, fermentation will occur in which the pyruvate will be converted into ethanol or lactate by the process of lactic acid fermentation or alcohol fermentation. Now let's look on the two stages of glycolysis. First is energy investment phase. Bear in mind why we call it as energy investment phase, because in this process we do not produce any ATP, but we will use ATP for the process to occur. So, the first step, glucose will enter the cell and thus it will get phosphate group from ATP and the ATP will become ADP. And the phosphate group attached at the 6 carbon of the glucose. Remember, glucose has 6 carbon skeleton. So 1, 2, 3, 4, 5, 6. Phosphate group from ATP attached at the carbon number 6. Therefore, it will give the name will be glucose 6 phosphate. And then what will happen? Isomerization will occur, which is that the internal arrangement of the glucose 6 phosphate will be changed, thus producing fructose 6 phosphate. So how does the internal arrangement looks like? So here is the structure of the glucose. The structure of the glucose, the carbonyl group is attached at the end of the carbon skeleton. However, internal arrangement occur. There is a changes in the internal arrangement, and thus the carbonyl group now is attached within the carbon skeleton of the fructose. So now glucose becoming fructose. Okay, and then we will get another phosphate group from the second ATP. Thus, the second phosphate group will attach at carbon number 1, and fructose 6 phosphate will become fructose 1 6 biphosphate, which is referred to two phosphate group attached at carbon number 1 and carbon number 6 of fructose. And then the structure of fructose 1,6 biphosphate is not stable, and then it will be split into two molecules of PGAL. Therefore, each of PGAL group will got one phosphate group. Okay, and PGAL can also change its internal arrangement, thus becoming glyceraldehyde 3 phosphate or dihydroxyacetone phosphate, DHAP. So from glucose to PGAL is the process of energy investment. So this is the more clear if you want to see more later. So in summary, energy investment phase use glucose to produce 2 molecules of PGL or DHAP or G3P, and it will use 2 ATP, thus becoming 2 ADP. Bear in mind, in this phase, there is no energy produced yet. Next is energy yielding phase of glycolysis. In energy yielding phase, so what will happen? The PGAL will receive phosphate group and attach at the other side of 2 PGAL. Thus, it will become 1,3-biphosphoglycerate because we have 2 phosphate group in each molecule of PGAL. And then it will give out 8 electron and hydrogen for a 2 NAD+, and NAD+ is reduced, becoming NADH. Since we have 2 NADH, since we have 2 PGL, therefore 2 NADH molecules will be produced. Next, 1,3-biphosphoglycerate will donate, each of it will donate phosphate group to ADP. Thus, ADP will become ATP, and 1,3-biphosphoglycerate form 3-phosphoglycerate, and then 3-phosphoglycerate will change its locations of phosphate group to the second carbon. Now the name is 2-phosphoglycerate, and then 2-phosphoglycerate later will undergo isomerization, and thus it will become PEP, or known as phosphoenol pyruvate. And then 2 molecules of phosphoenol pyruvate will remove its phosphate group, and the phosphate group will attach to ADP, becoming ATP, and it will become 2 molecules of pyruvate. So, remember, from 1 glucose we produce 2 pyruvate. We use 2 ATP, now we produce 4 ATP. And we convert NAD+ to become 2 molecules of NAD+ over here. So, in summary, okay, this is the more clear view, a clear picture of it. So in summary, energy yielding phase or energy payoff phase, PGAL converted into pyruvate, NAD+ is reduced, becoming NADH, and 4AD+ form 4 ADP, forming 4 ATP. So this is the whole process of glycolysis. I gave you 2 pictures here, so that easy for you to choose which one you. It's easier for you to understand. And this is how does the input and the output look like. During energy investment phase, we use glucose, right? So the glucose later will produce 2 pyruvate and also 2 molecules of water. We borrow 2 ATP and then we produce 4 ATP. So we have to pay back the ATP that we borrow. Now 4 minus by 2, the net profit or the net gain of ATP is 2 ATP. Meanwhile, 2 NAD+ has been reduced, becoming 2 NADH. So in conclusion, 3 most significant changes that take place during glycolysis, which is first, 1 molecule of glucose converted into 2 molecules of pyruvate, 2 ADP is converted into 4 ATP net gain, and 2 molecules of NAD plus is being reduced to 2 molecules of NADH. So I will give you the link of the video later of how does the glycolysis occur in cytoplasm. Next, link reaction or transition reaction. So the pyruvate from glycolysis will enter the mitochondrial matrix through the process of transition. So what will happen? Pyruvate will give it carbon atom and 2 atoms of oxygen, thus forming carbon dioxide. Now, 3 carbon molecules, it will become 2 carbon molecules. After the carbon dioxide is given off, NAD plus, 2 NAD plus molecules later will be converted to becoming 2 NADH. Okay, I forgot to put 2 over here, 2 molecules of NADH. And then coenzyme will attach to the remaining 2 carbon, which is known as acetyl, and thus it forming acetyl-CoA. Bear in mind, glycolysis produces 2 pyruvate. Thus, transition reaction later will produce 2 molecules of carbon dioxide, 4 molecules of NADH, and 2 molecules of acetyl-CoA. So this is the picture for you to understand more later. And next, the second stage in aerobic respiration is Krebs cycle, or known as citric acid cycle or tricarboxylic acid cycle. It is named after the Hans Krebs. Okay, so what will happen here? The acetyl-CoA from transition reaction will enter the Krebs cycle, and then this Krebs cycle involve an enzymatic reaction. It occurs in the mitochondrial matrix, and most of the reaction involve oxidation reduction reaction and also decarboxylation reaction. Okay, let's look more into Krebs cycle process. Krebs cycle process involve 8 steps. So this 8 step you have to remember. You must remember. Okay. So this picture only show one cycle or one turn of citric acid cycle. Okay. So what will happen? Acetyl-CoA have 2 carbon backbone, right? 2 carbon atom backbone. Okay. It will given off its coenzyme, and it will combine with oxaloacetate. Oxaloacetate have 4 carbon backbone, 1, 2, 3, 4, and thus it will become 6 carbon molecules, which is known as citrate. And then the citrate will remove water and accept water back, and this is cause the internal arrangement to change. So this process is known as isomerization. The internal arrangement of the substrate is changed, thus becoming isocitrate. Isocitrate is the isomer form of citrate. And then step number 3, isocitrate will undergo oxidation and decarboxylation, in which it will remove its carbon dioxide and thus an oxidation because it isocitrate remove its hydrogen ion, decarboxylation because isocitrate remove its carbon dioxide to becoming a carbon dioxide, and thus it will become alpha ketoglutarate. Now the carbon backbone got 5 carbon only. 1, 2, 3, 4, 5 from 6 carbon backbone over here. And NAD plus will undergo reduction process becoming NADH. After that, alpha ketoglutarate will undergo decarboxylation process again. It will remove carbon dioxide and it also undergo oxidation process because it donates its hydrogen to NAD plus, and NAD plus is being reduced becoming NADH. At the same time, coenzyme will enter and thus becoming succinyl coenzyme. In step number 5, coenzyme will be removed and replaced by phosphate group. And this phosphate group will attach to GDP, guanine diphosphate. Thus, it will produce GTP, guanine triphosphate. Guanine triphosphate is just a carrier of phosphate group, and then it will give the phosphate group to ADP. Thus, ADP will become ATP. And then, succinyl CoA become succinate. After that, succinate will undergo oxidation in which it will donate its hydrogen ion to hydrogen atom to FAD, and FAD undergo reduction process becoming FADH2. And succinate, after being oxidized, it will become fumarate. And then fumarate will become malate through the process of hydration, or known as additions of water. Next, malate will be oxidized because it removes its hydrogen atom, and NAD plus will undergo reduction process because it accepts hydrogen from malate, thus becoming NADH. And malate, after being oxidized, it will become oxaloacetate, which is got 4 carbon atom at the backbone. And then the cycle will turn again for the second time. Why citric acid cycle or Krebs cycle occur twice? It is because for first cycle we only receive 2 acetyl-CoA. You have to remember we have 2 molecules of acetyl-CoA. So we will need to have 2 cycle of citric acid. Therefore, from 2 cycle we will produce 2 ATP. From 2 cycle we will produce 6 NADH, and from 2 cycle we will produce 2 FADH2, and from 2 cycle we will produce 4 carbon dioxide molecules. So this is another example of Krebs cycle. So in conclusion, Krebs cycle require oxygen. That's why it is known as aerobic. Okay, so each turn will produce 3 NADH, 1 FADH2, and 1 carbon dioxide, 2 carbon dioxide molecules, and 1 ATP. Since the cycle turns twice because of 2 molecules of acetyl-CoA, so everything will multiply by 2. Then we will get 2 NADH, 2 molecules of FADH2, 4 molecules of carbon dioxide, and 2 molecules of ATP that being produced by substrate level phosphorylation. So this is the video of the Krebs cycle. I will give you the link later. So I think that's all for today. Thank you, class.